Hello, everyone, and welcome to the National Academy of Inventors ScholarShare webinar series. My name is Jade Stewart, and I currently serve as the director of the National Academy of Inventors. For those who may not be familiar, the NAI is a member organization comprising U.S. and international universities, government agencies, and nonprofit research institutes with over 4,000 individual inventor members, fellows, and senior members spanning more than 250 institutions worldwide. The NAI was founded in 2010 to recognize and encourage inventors with patents issued from the United States Patent and Trademark Office, enhance the visibility of academic technology and innovation, encourage the disclosure of intellectual property, educate and mentor innovative students, and translate the inventions of its members to benefit society. We offer a variety of programs for our members and partners, including our mentorship platform, which is commonly referred to as GAIN, the Global Academic Inventors Network, connecting students, researchers, and faculty from across the world. We also have our in-house multidisciplinary journal, Technology and Innovation, published 4 times a year. Also, our most recent launch of From Campus to Commerce, a video series featuring early stage innovation that takes place at our member institutions. Stay tuned for an upcoming episode featuring Dr. Katti, who is our guest speaker today. But we also have had a number of organizations and universities represented, including Beyond Meat from the University of Missouri, a plant-based protein alternative. We also offer 2 recognition and award programs, fellows and senior members. We are still accepting nominations for senior members and encourage you to submit an application or nominate one of your peers for these awards. Our organization is committed to using its platform to help minimize barriers and promote an environment that is inclusive for all, especially through these programs I mentioned. NAI is one of 3 inventor recognition organizations that partner with and have the full support of the USPTO. And today's fireside chat is titled Immunotherapeutic Nanomedicine Through Green Nanotechnology, Preclinical and Clinical Investigations. And I would like to introduce our speaker, Dr. Kattesh Katti. Globally recognized as the father of green nanotechnology, Professor Kattesh Katti is internationally renowned as a leader in the interconnecting fields of chemistry, radiopharmaceutical sciences, green nanotechnology, and nanomedicine. His focus over the past 3 decades has been on molecular imaging in therapy and oncology. He has also pioneered work in combining non-toxic gold nanotechnology with Ayurvedic holistic medicine. Dr. Katti's cancer therapy products and antibiotics are in use on patients today. He has also been recognized by the United Nations, IAEA, as the global expert in green nanotechnology, and has won many awards, including International Hervesi Medal Award, the 2016 Person of the Year in Science, as well as Fellow of the National Academy of Inventors. So please join me in welcoming Dr. Katti. Thank you.
Thank you. Thank you, Jade. Thank you, Jody. Thank you, the rest of the staff and leadership at the National Academy of Inventors for this great opportunity through this ScholarShare program where National Academy of Inventors is featuring discussions about latest technologies and how laboratory discoveries can be taken up all the way from ideas to inventions to patents, all the way to value-added products, perhaps for saving human lives, and many, many other applications in human walks of life. Today, I would like to take you through a journey of our approach to cancer therapy. I would also like to take you through our journey, how we started off with simple ideas to contribute to fundamental science and technology. And through collective collaborative efforts, how through our University of Missouri's Patents and Licensing Office, we have been collectively successful in identifying investment groups and corporations where our Patents and Licensing Office has been able to license our technologies, and the licensees have been able to translate our technologies into products. So that's one of the strong undercurrents of this great academy, National Academy of Inventors. So I've been very fortunate that I have had this invitation to speak with all of you today. Well, a quick intro about what we do here at the University of Missouri. There's a very powerful culture here, culture of clinical translation. As you can see here, there have been a number of products. A vast majority of them use radio labels for both diagnostic and therapeutic applications. As many of you know, University of Missouri houses the world's largest nuclear reactor at the university level, and that has catalyzed our clinical translation efforts tremendously. And today what you will see are the latest developments where we are able to apply nanotechnology, principles of nanotechnology, to make the drugs, cancer drugs, more effective, both for diagnosis and therapy. A quick outline, snapshot about what I'll be doing over the next 40 minutes or so. I will describe to you, as I said, nanomedicine approaches in cancer therapy. We'll discuss why it is absolutely imperative to be thinking about new therapeutic approaches. And then we'll define what, according to us, is green nanotechnology. And then we'll justify that this is a very constructive way forward, whether you're using nanoparticles for medicine or perhaps for any other applications. Then we'll probe into the mechanistic details about how these gold nanoparticles, the ones that we have discovered, they are not just simple cytotoxic agents. They have immunomodulatory role in that once administered, they provide the body the strength to fight the disease. So it boosts the immune responses within the body. That's how we are able to show that we can effectively treat deadly cancers. And then I'm going to bring to you this very serendipitous discovery that we have been able to connect the discoveries, what we have made, to some of the Ayurvedic medicine approaches which are being used for over 5,000 years in different parts of the world, especially the Southeast Asia, India, Indian subcontinent. Lastly, as all of us recognize, no talk under the circumstances that we live in would be complete without alluding to some implications, how this science, what we have developed, has really practical implications in developing antiviral agents based on silver nanoparticles. Well, I will not spend too much time here giving you an introduction about how deadly this disease is. The singular disease, as all of us know, affects every nook and corner of the world. Cancer kills more lives compared to tuberculosis, AIDS, and malaria combined. And this is also referred to as an economic killer because it takes away 1.5% of the total global gross domestic product. The prognosis looks very bleak. We have many therapies. We have chemotherapies, new generations of immunotherapies. We have radiotherapies. We have varieties of armamentarium of therapies for combating this disease, but still the prognosis of this disease doesn't look good. We're going to see substantially more number of cases of cancer patients across the world. It's not because our quality of life will be compromised on the quality of life in the near future or so. It is simply because our ability to detect the diseases, the diagnostic agents are becoming more and more smarter. That means that we're going to see many cases of cancer patients across the world. So the problem is real. It's not something that we don't have it here or it's more somewhere else. It's there everywhere. So we need a collective effort for that. So what's going on right now? The status quo, if a patient is presented to a doctor, and if the patient is diagnosed with certain types of cancer, breast cancer, pancreatic cancer, glioblastoma, or prostate cancer, there are effective therapies. There's no doubt about that. The two main therapeutic approaches, despite all the developments, the two main workhorses for therapy when it comes to treating cancer patients are the chemotherapy and the radiation therapy. Of course, as I said, we have the latest advances through immunotherapy, CAR T-cell therapies, biologics, and so forth. But these two are still the workhorses for handling the care and management of cancer patients. But the chemotherapy and radiation therapy, as great as they are, they also pose the role of a double-edged sword. That's because once administered, once patients are treated through these two modalities, it leaves a lasting effect on the patient. A number of times, this approach actually makes the disease resistant to future therapies. And that's so because these therapies activate a master signaling factor, cell signaling factor, that's called as the NF-kappa B. And this master signaling factor has the ability to activate many, many downstream signaling pathways. In other words, this signaling factor sends messages across the body, okay, to really activate the progression of the tumor, metastasis of the tumor, and so forth. So on the one hand, we are trying to use these therapies to treat cancers, but it leaves that lasting side effect, which oftentimes can be deadly. So that's why we need therapies that target or deactivate this signaling pathway. That's some aspect, along those aspects, we are going to look at how nanotechnology can help us. Those of you within the audience who are experts in nanomedicine, I will be preaching to the choir. You already know, as we go from bulk materials to nanomaterials, we reduce the size, but we gain continuously in something. We gain in terms of the surface area. If you have nanoparticles of gold, you have fewer atoms of gold in bulk materials, but you have hundreds to thousands atoms of gold on the surface. What do we do with them? We can use individual atoms in many, many practical ways. Those atoms can be used as points or as suitcases in a simpler way to put diagnostic probes, therapeutic probes. The whole idea here is to enhance bioavailability, to make the therapeutics or diagnostic site-specific. Site specificity is an aspect based on size. We're going to come to that in a moment. But amplification of therapeutic or diagnostic payload, it's not a wish list. It's based strictly on the rules of physics. If you have so many atoms, each of the atoms can be tagged on or conjugated through covalent interactions with either the diagnostic or therapeutic or agents which have dual diagnostic and therapeutic power for theranostic applications. All of those we have been talking for almost now 20 years. Now we are seeing some practical implications that we can actually make use of the principles of nanotechnology for smart medical applications. The focus of today's talk is gold. There are not too many questions. Why gold? As you can see from the famous philosopher. But gold is attractive for nanomedicine because it's the only element in the periodic table which remains unoxidized at the nanoparticulate level. And that's because it has very favorable redox properties, and the redox properties in turn are related to its periodicity. All of us know that. Gold is also very attractive from where we are at the University of Missouri, as I was telling you. We have the world's largest university-owned nuclear reactor, and we have utilized that reactor in a very fruitful way to develop cancer therapy, cancer diagnostic agents for saving human lives across the world. And one of the areas where we have focused over the last 2.5 decades, almost 25 years now, is to transform normal gold into radioactive gold. Why do we do that? Radioactive gold gives you 2 isotopes as shown here. And these isotopes have favorable beta emission for cancer therapy. They also have gamma emission for cancer diagnosis, which means that singular nanomedicine agents, which are capable of doing dual therapy and diagnostics, can be realized, actually. It's not a lofty ideal. So theranostic applications of these nanoparticles, I'm not going to discuss today on the radioactive nanoparticulate subject matter. However, I'm going to use some of the radioactive nanoparticles to show all of you how effectively we can calculate the amount of gold per organ. If it is a tumor, we can use radioactivity through the scintigraphic imaging as well as scintigraphic counting techniques to quantitatively analyze the biodistribution pharmacokinetics of gold-based nanomedicine agents. Well, as I was telling you, I'm going to be using this interesting term, green nanotechnology. Green is a cool term from many points of view. Green symbolizes clean water, clean air, natural forests, rainforests. Everything is great for the human civilization. And we're going to use somewhat similar principles here in the context of transforming gold or other metallic precursors into their corresponding nanoparticles. The simple chemical reaction, as all of us know, if we have to convert gold precursors or silver precursors into nanoparticles, we need chemical reducing agents, which means that we need reservoirs of electrons. We can use sodium borohydride, hydrazine. That's, in our opinion, may leave a toxic trail behind, not just the production of nanoparticles, but the production of those chemical reagents and so forth. But there's a more practical and attractive way, which we discovered almost 25 plus years now, that we can use those abundance of electrons that we get from these powerful antioxidants, which are there in many of the plant species, such as tea leaves gives us epigallocatechin gallate, mango gives mangiferin, grapes, we have resveratrol, cloves, we have eugenol, we have a host of these plant species. And look at the electron density or the plethora of electrons that we can harness from these plants. And then when they interact with gold precursor, we reduce gold from +3 oxidation state, and that's how we produce nanoparticles. And the particles so produced, generally speaking, they don't require an additional encapsulating agent, because as we know, because of the surface activity, because of the surface atoms, very often we need to stabilize these nanoparticles with chemical entities. So in these cases, the powerful hydrogen bonding that creates a very thick corona helps us in stabilizing the nanoparticles, helps us in formation of the nanoparticles. There is an added bonus, and those coronas very often help us in transforming these nanoparticles to be disease-specific or tumor-specific. All of that we're going to see. And this is how those nanoparticles look like. So we have these nanoparticles with a very thick corona of mangiferin, and we have been researching on a number of different phytochemicals, as you may have seen our publications over the last 20 plus years. We like the pharmacomotif of this structural motif of this polyphenol. It is a polyphenolic structure conjugated with glucose moiety, as you can see here. The fact that there is glucose, it allows us to enhance the uptake of these nanoparticles within the tumor sites, because tumor cells, the mechanism of tumor metabolism, they metabolize sugars much better than many other chemical species. And that plays a very significant role, that has played a very significant role in the choice of this particular phytochemical in our nanoparticle formulation, as well as the application of these nanoparticles. As I said, we always make the corresponding radioactive nanoparticles, which have applications in radiotherapy, both for diagnostic and therapeutic applications. But when we are using just non-radioactive species, the radioactive counterparts help us understand localization, the quantification of these nanomedicine agents in vivo, in the body. And this is how the corona looks like. It's a very thick network of hydrogen bonds, very strongly held both through sigma pi interactions, as well as electrostatic, as well as hydrogen bonding interaction. This is a very powerful corona, and we see incredible in vitro and in vivo stabilities. When you're talking about nanotechnology, application of nanoparticles for medicine, we need to be aware of the size. Why size is important? As all of us know, normal cells have no vasculature. They're very tightly held. Tumor cells provide us a leaky vasculature. That means we have a porosity of about 150 to 300 nanometers. As long as we keep the hydrodynamic size lower than this porosity, we have a good chance to penetrate the tumor vasculature, push therapeutic or diagnostic payload across the tumor membrane. This is only one of the mechanisms how we can localize these nanoparticles site specifically around the tumor region. There's a more sophisticated way wherein we have discovered that mangiferin has selective affinity to laminin receptors, which are overexpressed on prostate, breast cancers, colorectal cancers, pancreatic cancers, and so forth. And those laminin receptors, once we encapsulate these nanoparticles with laminin receptor-specific mangiferin, we see these types of endocytosis. There is an abundance of nanoparticles in each of these cells. In order to show that this is not by accident that we are seeing these nanoparticles within the cells, we block those laminin receptors using laminin-specific antibodies. Just one example I'm showing here, ABLR antibody. When we block those receptors, and then when we try to incubate mangiferin nanoparticles onto these cells where the receptors were blocked, we clearly saw hardly any nanoparticles getting internalized into the tumor cells. As you can see, almost all the nanoparticles remain outside the cellular membrane, suggesting to us that the endocytosis is indeed mediated through laminin receptors overexpressed on these types of tumors. The propensity with which these nanoparticles are seen inside the cells gave us some confidence. We did biodistribution studies. In this case, we used the corresponding radioactive nanoparticles. We introduced it intratumorally because intratumoral delivery is important, because primary tumors, particularly of the prostate, in the case of castration-resistant prostate tumors, where the majority of the people have no luxury of surgical resection, it is the primary tumor that keeps spitting out the tumor cells, and they're recruited into the bone marrow. That's how the tumor spreads across the body. There are very few therapeutics right now that are able to penetrate the tumor interstitia. So that's why we thought that these are attractive for addressing solid tumors. That's why we introduced these particles into the solid tumor, and then studied if there is any lymphatic drainage of these nanoparticles. As you can see here, these particles are held, retained very well within the tumor, very limited lymphatic drainage, and that translated into really spectacular therapeutic efficacy curves. It's dose dependent. We're able to control prostate tumor in prostate tumor bearing mice, metastatic breast tumors in breast tumor bearing mice. Again, great dose dependency. At this stage, we started asking those questions. We know that mangiferin has immunomodulatory power, but once it goes onto a nanoparticle, there is no guarantee that we didn't know whether it retains that immunomodulatory power. Not only that, we also wanted to take advantage of the synergistic advantages here of mangiferin with gold, because gold nanoparticles are inherently angiogenic. So that was the whole idea. That was the hypothesis. Then we started looking at the immunomodulatory power, the master regulator. We tested NF-kappa B upon mixing with the tumor cells. As you can see here, we first enhanced that NF-kappa B by stimulating with TNF-alpha. As you can see here, these gold nanoparticles actually target NF-kappa B. We can see a substantial reduction in NF-kappa B levels in tumor cells that were incubated with mangiferin gold. Mangiferin does to some extent, but mangiferin alone has not found applications in cancer therapy, as all of you know. Once mangiferin is there in the body, it is metabolized so fast. Its bioavailability is a big question. With mangiferin encapsulated nanoparticles, we are seeing that we can enhance the bioavailability. Most importantly, because of the compacted nanoparticulate size, we can enter the tumor cells and initiate these immunomodulatory characteristics. Then we quickly looked at their propensity with prostate tumor cells, very, very selective. You can see almost every prostate tumor cell having abundance of this mangiferin nanoparticles. Again, laminin receptor specificity. At this stage, we also wanted to check if these particles enter the macrophages. Why the macrophages are important? Because we have to recognize that just killing tumor cells doesn't do the job. The tumor stroma has macrophages as well as these varieties of other non-cancerous cells. They are recruited away from the tumor, and it is the macrophages that result in pro-tumor phenotypes such as M2, and it's the macrophages that will elevate the levels of IL-10. So drugs incapable of targeting the tumor microenvironment, all of us know, those drugs treat the tumor to some extent, but the untreated tumors become resistant to future therapies. So that's the whole problem, and then the tumor therapy must address ability to target macrophages. If the therapeutic agent fails to target the macrophage, we know that from varieties of scientific evidence and clinical evidence, they don't do any good in terms of therapy as a whole. What you see here is another example to check if these particles target the macrophage. We did the biodistribution, whole body biodistribution. There's a substantial dose that is accumulated within the spleen. This is where we thought that there is a targeting pathway, targeting of the splenic macrophages. And that's when we decided to really go after the macrophagic cells. What we did here on this slide, we basically incubated prostate tumor cells with macrophage cells. And these macrophage cells, they are always pro-tumor, okay? They're of the M2 type of macrophage. By incubating prostate tumor cells with macrophage cells, you can see there's a proliferation of prostate tumor cells, which is expected. In a separate experiment, what we did, we incubated macrophage cells with mangiferin gold nanoparticles. The hypothesis here was that mangiferin gold nanoparticles will re-educate these macrophages from being pro-tumor into anti-tumor. They will re-educate, they will transform these pro-tumor M2 macrophages into M1 anti-tumor macrophages. If these mangiferin nanoparticles are able to target the tumor microenvironment, if they're able to target the macrophages. That's exactly what we saw actually. The macrophage cells pre-treated with mangiferin gold, when those cells were incubated with prostate tumor cells, we saw a substantial reduction in the proliferation of prostate tumor cells. It's a great evidence that it's a very reliable biomarker to conclude that our gold nanoparticulates encapsulated with mangiferin they exert immunomodulatory influence on the tumors. They transform pro-tumor macrophages into anti-tumor macrophages. And at this stage, we had pretty much everything. We had proven our hypothesis. We had scaled up the synthesis. We had done reproducibility by collaborations with various different labs. And then we approached the patent office at the University of Missouri. They did a remarkable search for prospective investors. And then they found a licensee. Licensee was very excited about the process because it's a great example of a zero carbon footprint process because there is no gas that comes out. There is no toxic material that's within the reaction scheme. And they liked it, actually. They felt that they can scale it up. And they indeed licensed the technology. They set up the manufacturing plant in India. There's the largest green nanotechnology based manufacturing plant in the world that is there in Chennai. Those of you who would like to take a look at it, contact me. I will direct you people there. Then we started talking about clinical translation, because when you have an investor, they invest, then they really look at return on investments, which is the right thing to do. So the production was complete, toxicity studies were done. Then they started approaching Indian regulatory agency, the Ayush, which is a regulatory agency for approving Ayurvedic medicines. The connectivity between what we have done to Ayurvedic medicine, I will show you in a moment, but they got the approval. Through that approval, they conducted pilot clinical trials, and the data is shown here. These nanoparticles actually are great. Initial clinical trials were done to test their efficacy as an adjuvant therapeutic agent. These are pre-treatment tumor burden, the median tumor burden. 90 days post-treatment, you might say, yeah, there was 6 to 8% tumor reduction. It's important to recognize that these are metastatic breast cancer patients. They have very limited therapeutic options. But this effect looks highly significant when you compare this group of patients, human patients. These groups of patients, they received just the chemotherapy. They did not receive the adjuvant, manganese gold-based therapy. Just so that these are trade names. The products are sold under NanoLife. DNA Stana is the trade name of this manganese gold nanoparticle agent. And anyway, when you compare these two, patients who were given only the chemotherapy, the tumor burden either doubled, tripled, or quadrupled. The quality of life of a majority of the patients was miserable. Quality of life in terms of the body weight, eating habits, overall moods were normal here. And it's just the beginning. There are more clinical trials in progress. So far, we have seen that the immunomodulatory power of this agent appears to really help chemotherapy in ways we haven't seen, we rarely see, in this modality. You might call it an isolated case, but now that the word is out that this agent is helpful in treating cancer patients, one of the hospitals approached the Indian company. They got hold of the medication, and the patient was administered. This is the pre-treatment. You see lots of lesions. This is a stage 4 ovarian cancer. 90 days post-treatment, almost all the lesions have disappeared. You can see evidence is very clear. So it's clearly showing efficacy. It is showing efficacy because it targets the tumor microenvironment, promotes the formation of IL-12 cytokine. It suppresses IL-10, which is the pro-tumor cytokine. It promotes IL-12, the anti-tumor cytokine. So all the goodies what you need it to be an immunotherapeutic agent that are there in this new modality. As we saw success with manganese encapsulated gold nanoparticles, we now have produced a library of these nanoparticles. We are investigating immunomodulatory power of e.g. CG encapsulated nanoparticles, resveratrol encapsulated nanoparticles. So what I'm trying to tell here, one size doesn't fit all, we know that, but this approach of green nanotechnology where we can encapsulate tumor-specific phytochemicals allows us to develop a library of immunomodulatory therapeutic agents. Let's quickly take a look at the history. What's going on with metallic gold in the context of its use for medicine throughout human history. Gold in various formulations has been used, as I said, over 5,000 years ago by Ayurvedic medicine in India. It was used, it's still being used by the Chinese, by the Greeks, by the Romans. So what we have done so far is encapsulating or using electrons from phytochemicals and creating a process to produce phytochemical encapsulated nanoparticles. If you look at Ayurvedic medicines, they have been doing like that for about 5,000 years without understanding the science. There are problems in these formulations because their way of making gold nanoparticles, they don't get reproducible results. And their particles, they're not 100% nanoparticles. They have 1 to 2% nanoparticles, which are of the right size. Majority of their formulations are macro in size. No wonder their results are not reproducible. And very often you can term their results as anecdotal. With our approach, we have made the approach non-toxic because the traditional gold-based medications, they use mercury amalgam, they have impurities of lead and so forth, but we have refined this approach in ways that we have gold nanoparticles and the phytochemicals. We have done extensive toxicology analysis in different animal species, and now we have doses, therapeutic doses at which we see no systemic toxicity. So it's all going on. At this stage, what we are doing is reverse engineering it. We know a lot from history. People have used gold formulations without knowing the science. We developed the science through green nanotechnology, did reproducible formulations, developed industrial scale manufacturing, understood the efficacy in animals. Now we know the immunomodulatory mechanism. So started with humans. Now we have done all of this cycle and finally going back to humans with the scientific rigor, scientific reproducibility, all of that. So that's why I call it as a great way of reverse engineering, connecting our science to the age-old holistic Ayurvedic medicine. And this is when we sought whether we can get an approval by the United States Patents and Trademarks Office. They did approve this nano Ayurvedic medicine trade name for various cancer therapy and antibiotic and antiviral products for these formulations. So we have agents for use for breast cancer, for prostate cancer, and for other cancers because it targets NF-kappa B, which is a signaling factor that's prevalent in many of the inflammatory diseases and disorders such as diabetes, arthritis. The medical doctors in India are really exploring with great data already on the applications of these types of medications for diabetes and other diseases. WHO, as all of us know, and in their forecast, they're telling that over 80% of the world's population would love to have something that's green, something that's holistic. So Ayurvedic medicine, when approached through nanotechnology, appears to provide us a great future. Yeah, last 10 minutes, maybe less. So far about gold. Silver, well, let's talk about silver. Silver, historically, it's the only element in the periodic table which has great antibiotic properties, antiviral properties. But metallic compounds have done very little in terms of efficacy. Metallic compounds often tends to be way too toxic. This is where we hypothesized that we could create biocompatible silver nanoparticles and then test their efficacy for antibiotic and antiviral applications. We have a host of silver, biocompatible silver particulate-based drugs that we're able to use to destroy such deadly bacteria such as MRSA, staph, and so forth. But today I'll just share with you silver nanoparticles encapsulated with glycoprotein, how effectively they target the viral machinery, so that we can begin to use these types of nanoparticles in our collective efforts to control and neutralize this COVID-19 pandemic. The hypothesis has been that the silver nanoparticles encapsulated with glycoprotein, because of the protein-protein interaction. Glycoproteins interact with the spike protein of the COVID, and this interaction will facilitate transfer of silver nanoparticles into the viral machinery. But not only that, if we can saturate the viral surface with glycoproteins, it would inhibit the viruses from entering into the host cell. Thus, there's a great chance that we can eliminate the viral infection throughout the body. With that hypothesis, we developed these nanoparticles. Now they have been scaled up at the industrial scales, and the approach what we have taken is shown here. If the virus is left alone, they will infect, they will enter the normal healthy cells as shown here. But if the viral particles are blocked by these glycoproteins on silver nanoparticles, the glycoproteins not only stop the viral entry into the healthy cell, but the glycoproteins interaction with the virus surface will allow silver nanoparticles to get into the viral machinery. And that is the direct effect why we are seeing a really great viral COVID-19 neutralization efficacy. We are right now at 99.68%. We are on our way to get to 99.9. Some of the recent results do show that we can get to that level. Just wanted to show you this one. The effect of the virus, effect of the nanoparticles, which is important to recognize, we see no toxicity to the normal cells. So cytotoxicity is not an issue here. These particles are highly toxic to the virus. The toxicity measurements have been done for these silver nanoparticles. According to OECD guidelines, we have seen no toxicity even at 300 to 500 milligrams per kg of body weight. Almost ideal. So I would like to see from our group, from other groups, silver nanoparticles as a way to treat these deadly viral infections, because there have been many, many different approaches. I strongly believe that nanotechnology through silver nanoparticles has an important place in our collective efforts to control this pandemic. With that, I would like to share the products that have come out through our patents, which have been licensed to this company called DNA in India. They have cancer therapy products. They have products for infections. They have products for arthritis. This is a product. This is a hand sanitizer which doesn't use any alcohol. The viral effect, antibacterial effect is based solely on silver nanoparticles. So there is no toxic organic chemical in this product at all. So they have approval in India, in Africa, all of Middle East. They have set up an office in the US recently. Soon they will have these products available for use within the US. With that, I would like to thank lots of people here. The work I presented, it's a work that has spanned nearly 3 decades. People ask me, Kattesh, how do you do this? What is nanotechnology? Nanotechnology doesn't just fall from the heaven. I wish it was like that. It's a combination of different disciplines: physics, chemistry, biology. In our case, isotope production experts. We need tumor biologists, mathematical modeling experts, synthetic chemists. Everybody has a place in nanotechnology. I'd like to thank all these students. One thing that's common with every one of these students in the last 30 plus years is that every one of them turned out to be significantly smarter than me. That's why we have been able to do all of this work. Thank you, all our past students, current students. I would like to thank my wife, who has been contributing to this science in ways hard to describe, my life partner as well as a partner in my career. International collaborators, great team of collaborators, faculty collaborators with a great degree of specialization. Again, a National Academy of Inventors for this opportunity. Funding agencies, I'll come to that in a moment. It all started with funding from the National Cancer Institute. We learned a great deal about quality control for nanoparticles through our collaborations with Nano Characterization Laboratory. IAEA has been very helpful. Brazilian CNPq through Professor Adimar, whom I would strongly recommend as one of the ScholarShare speakers in the near future. Again, lots of these agencies, my institution, which has been extraordinarily cooperative and helpful in every aspect. And I know you will have some questions, comments. Now is the time for that. For those of you who could not be here or who may have to leave now, here are my email coordinates. Send me an email if you have a comment or a question. I know, Jade, I took a few extra minutes. Now is the time to get questions and comments. Thank you, everyone. Thank you, Jade. Thank you, Jody. I will stop now.
Thank you, Dr. Katti. That was a very enlightening presentation and a job well done. So yes, if you do have any questions, please feel free to use the chat function or the Q&A feature on Zoom. So the first question I have queued is, why do you think gold is so unique in nanomedicine?
Yeah, it's a great question. There are many elements in the periodic table. Of course, there are particles of cadmium, selenium, carbon. Everything is important, no question about that. But when it comes to medicine, we know gold under certain doses is well tolerated in the body. Gold is important from the chemistry point of view. It remains unoxidized at the nanoparticulate level, which is extremely important. In my opinion, there is a comfort zone here. Gold, as I was telling you, has been used for over 5,000 years. We haven't seen major problems as long as you control the dose, as long as you pay due attention how you formulate gold-based drugs. And that's the reason.
Great. The next question I have for you is, can any of your discoveries be used to neutralize COVID-19, the deadly virus?
Yeah, I would say an emphatic yes. As I was telling you, there have been many discoveries we have seen from companies across the world, including the US, Europe, Asia, and whatnot, in our collective efforts to control and destroy and neutralize COVID-19. Silver, in my opinion, we haven't paid much attention to that. It gives us a very powerful tool to destroy the virus. Why we haven't done that? Because as I was telling you, much of our effort over the last 50, 60 years, all of that was focused on using silver-based compounds. But now with the advent of nanotechnology, I'm very confident that we and others will develop antibiotics and antiviral agents, including anti-COVID agents based on silver nanoparticles.
And then the last one, again, if you have questions for Dr. Katti and we run out of time, feel free to email him or email us at NAI. But this last question for you before we close out the session is, when do you think patients can access your products of Immunomodulatory nano Ayurvedic medicine here in the US? I'm also a huge fan of Ayurvedic medicine, so this question will resonate with me as well.
Yeah, Jade, that's a great question. We have been getting those questions either through email or telephone calls to my department almost on a regular basis, because people are excited about holistic approaches to treating different diseases, particularly cancer. So the answer to that question is the company in India, they do have a global presence. They recently established a unit here in New Jersey, and they're talking with Ayurvedic doctors within the US and North America. Right now, if a patient really needs, on a compassionate basis, they're able to ship under regulatory approvals to those patients. But soon it will be made widely available so that doctors can prescribe those medications.
Well, awesome. That sounds very exciting. And I'm super happy that we have you under our wing as a fellow of NAI to keep track of all of that activity. So I do want to thank you again for your time this afternoon and for a riveting presentation. This will be made available for future watch, available on NAI's YouTube channel, perhaps in the next week. And we'll make sure that link is disseminated to you, Dr. Katti, but to others who are on this call. I did want to acknowledge that we will have another webinar next week, Wednesday, October 14th, with the Undersecretary for the United States Patent and Trademark Office, Director Andre Yanku. And it will be a fireside chat moderated by our very own NAI board member, Karen Berg. And we invite you all to participate and attend that session. Again, it's October 14th at 3 p.m. next week. And with that, I again want to thank you, Dr. Katti. We will, of course, always be in touch. But again, if you do have other questions for him and want to learn more or have any information or additional information from this presentation, please do utilize his email address and webpage provided on the screen in front of you.
Thank you. Excellent. Thank you, Jade, and thank you, Jody. Thank you, everyone at the NAI. I would like to thank all the participants. As Jade said, I'm sure you'll have more questions and comments. Feel free to send them to me through email. Again, thank you all.